All right, so thanks everyone for coming to the first session of the afternoon. Our first speaker is Liset Menendez de la Prida from Instituto Cajal, and she'll be talking about high-frequency oscillations. So this session is essentially a combination of mechanisms and introduction to seizure prediction and devices for epilepsy. And our third speaker, Greg Worrell, I'm trying to track him down. Oh, there he is. Yeah. All right.
Thank you.
All right, cool. Thank you.
So, thank you, Levin, for the invitation. Can you hear me well? It's okay. So today I want to talk a little about normal and pathological high-frequency oscillations. It will be very general talk, an introductory talk, to try to discuss with you some of the basic elements that relate with this topic. And before going with the outline of the talk, most of what I will talk today is reviewed in this paper that we just published in Journal of Clinical Neurophysiology. This is the result of the last international workshop prediction, seizure prediction conference last year in San Diego, so it can be followed there. So first we will discuss about HFO, whether they are an oscillatory band or a broad frequency band, whether they reflect or represent an isolated event per se, or in contrast, there is some spectral leakage in that characterize the way in which this oscillation can be recorded. We will also talk about how they can be detected and the comparison between microelectrodes and macroelectrodes, and whether the appearance in micro and macroelectrodes will be the same or not. We will talk about bipolar and referential montage, how they could affect the waveform and the shape of the oscillations, or even create spurious high-frequency oscillations that are no longer present in the recordings. We will briefly talk about filtering, but not that much. To quickly go into what is physiological and what is pathological. This is a major question for us as neurophysiologists to try to understand whether there are specific features in the in high-frequency oscillation that can eventually let us to classify them as physiological or not. This is extremely important for clinical application, because you may be informed about whether a particular brain territory is producing normal or pathological form of activity. And finally, because we are also discussing about technological aspect, I will give a couple of slides to discuss whether we can modify HFO acting locally upon the mechanisms. Because evidently, and the data that I will present today is very basic and preliminary, it can be used, the tools can be used to try to assert a more specific action on this. So going to the point, high-frequency oscillations are activity recording in a type of brain activity at very fast frequency that can be recording with different settings. But brain activity can be recorded at the level of the surface with microelectrodes. But basically, we should try to understand that what we are recording in the surface near the sources can be related directly with the basic processes underlying spiking activity and neuronal firing. So that very wide char waves recording at the scalp can be directly related with a sort of gamma oscillations, activity that can be present in the local field potential that relate directly with the neuronal firing. Now, fast ripple is one example of this complex phenomena. And it was a milestone in high-frequency oscillations related to epilepsy. And it's now more than 20 years that Bryan and Engel sticking electrodes, microelectrodes, in the brain of epileptic patients, and they record this kind of activity when they recorded with microelectrodes in a referential montage that were pretty similar in appearance that what Bragging knew about ripples in the normal hippocampus of the rats. These ripples were typically recorded near the cellular layers and were associated with high-frequency cycles between 100 and 200 hertz that comes transiently during particular periods of activity in these animals. In epileptic patients and later in epileptic rats, they also describe a little more faster events that usually invades more than 200 hertz land. And because the similarity between these events and the ripple-like events with those recordings in the animals, in the rodents, they just term them fast ripples. So fast ripples resemble high-frequency hippocampal ripples, and therefore they were termed in this way. Later on, we have been struggling to try to understand whether they are a distorted version of fast ripple, which is the concept that I try to explain with the data that we collect in my lab. So going to the point whether ripples, fast ripple, or these high-frequency oscillations are an oscillatory event or are a frequency band, if you take a close look to microelectrode recordings in healthy rodents, in healthy rats, here I show 3 simultaneous microelectrode recordings in the hippocampus of a normal rat, and you can recognize that there is a large complexity of phenomena occurring spontaneously in these animals when the animal is immobile. So here we were targeting the CA1 cell layer, and you can recognize clear ripple-like activity that can be recorded simultaneously in different electrodes, which are separated about 1 millimeter each other. But also, you can clearly recognize other components, like, for instance, a fast run of gamma activity that is present in this particular microelectrode, but not really in this one, where it is more slowing down the activity. You also recognize individual action potentials, which belongs from a single cell that is firing just at this moment. And still, you can recognize small spikes here that we roughly term multi-unit activity because we cannot assign a particular spike to a particular unit because it is not reproducible and you can identify one firing after the other. So all these events can be collected together, and when you use microelectrodes in the hippocampus of normal rats, all of them can be collected and termed high-frequency oscillations because they are all above 80 hertz band. But they actually reflect a constellation of events. They don't reflect a single separate event. So for an electrophysiologist, a basic electrophysiologist, this is a ripple. But these will be gamma activity. These will be firing of individual action potentials, and that will be multi-unit activity. And all these different processes will impact onto the spectrum. So now if we take these 2 electrodes, a simultaneous recording from 2 microelectrodes, and we estimate the spectral, the time frequency spectrum of the different events, you can note how the dominant component actually slow down. Here is a particular pattern that is recording in the normal rats is ripples coming in as a tandem, like 2 or 3 together, and after that they typically come in association with a fast run of gamma activity and eventually other components that could appear. So we have 1, 2, 3, 4, and 5 sharp wave ripple events here that can be recognized. together with multi-unit firing and gamma activity. And the spectrum is rather complex and depict that evolution. And actually, if we take a close look here to this event here, we can see how in this electrode, the oscillations is associated with clear local field potential cycles that can be identified. And a neurophysiologist will tell you that these are the classical ripple in a normal rat. But can be also appreciated here very fast deflection, which correspond to individual action potentials that fire together from many cells just in the throat. And when many of those are tightly synchronized together, they shape very spiky the waveform, and therefore they contribute to a more larger, faster components of the event. In this electrode, those local field potential oscillations are not so much visible as here, but still few cycles can be recognized. But the contribution of multi-unit activity is more bigger, and it is reflected like a leakage at the level of the spectrum. So you can identify discrete events in the local field potentials of using microelectrodes or normal rats, and these discrete events are what we typically call ripples. But there are also different processes, isolated spikes, ongoing oscillations, multi-unit firing, that are contributing to the spectrum. So the spectrum of high-frequency oscillatory activity is reflecting a very complex sequence of events. But for a neurophysiologist working with Rodin, is very typical to try to focus in a particular state where we know that ripples are producing, like immobile or slow wave asleep. But the animal is also walking around, and when the animal walks around, what you typically get from the stratus radiatum and pyramidale is this theta activity. And on top of that theta activity, you record gamma activity, which has a phase relationship with this activity. And at the level, simultaneously, at the level of the molecular layer, deep in the hippocampus, faster components are coming. So if you take the spectrum along the death profile from the stratus pyramidale to the hilus, you see that the contribution of high-frequency activity, even in a condition where typically ripples, isolated discrete events are not present, is very complex. And therefore, it is not only what type of event you are recording from which cellular lamina you are recording. So that the complexity is not only in time, but also in space. And I mention this because when you are working with human settings, you don't have the possibility to be so precise to know exactly where your electrode is. But in any territory, neural territory, all the activity will be contributing to a very high frequency band, and will complicate the interpretation. So if you compare ripples recorded in normal rats with ripples recorded in epileptic rats, and you look at the frequency components, what characterize the activity in epileptic rats is that where ripples, physiological ripples, are typically more characteristic of low frequencies between 80 to 200 Hertz, you can have high frequency components that can be detected as bumps in the spectrum. And this is another factor that can help to identify whether you have a particular transient event which is associated with a truly local field potential oscillation, because it's associated with a bump in the spectrum. So power bumps, an increase in power within a particular frequency band, can indicate a potential short-lived HFO event that can be a discrete event that can be identified and not resulting from multi-unit firing, which will be more related with a spectral leak in the spectrum. HFO are also associated to and recording in epileptic rats. This is a recording from epileptic rats, recorded in association with interictal spike. This is a very sharp interictal spike recorded simultaneously in all the layers from the CA1 to the hilus, in the dentatic gyrus. And high-frequency oscillation, similar to ripples, are recorded near CA1 just before. And if you estimate the frequency component, it will not make this oscillation different to a ripple, to a physiological ripple. This is one example, and in the same animal, this will be another example. But this one will be more disorganized, and it will contribute to a very fast frequency band, something that never happens. So high frequency from high frequency oscillations is a relative term, as HFO encompasses a range of cellular and synaptic processes that contribute to a very wide frequency band from 80 Hertz to up to 800 or whatever you record from your filters. So how this looks like in recordings from micro and macroelectrode recordings. So the work by Worrell here has been fundamental in trying to relate with these typical electrodes using in clinical settings, macro and microelectrodes that can be associated, whether the events that can be recording on the tip can be related with events that can be recording on this microelectrode, which are a little far away from the sources where these microelectrode recordings were present. But if you take a close look to the data, and you see that how in the macroelectrode, which are averaging the activity of larger territories, and eventually the relationship between these local sources and the macroelectrode signals is not so straightforward, and we still need to understand. As you can see, the frequency components in the microelectrode recording are much more complex and much more diverse than those recording in the macroelectrode. And it can also impact the way in which you record from these micro and macroelectrode recordings. So therefore, the appearance of the event may differ between these two. Now, for a basic neurophysiologist, this means not that much, because we will fail to recognize typical hallmarks of activity that we can clearly recognize in the microelectrode settings. And not only that we are more expert in recognizing this signal, but also that we have been learning for a lot of work on how to relate specific waveforms here to the basic cellular processes that are present and distorted in the cases. So using this microelectrode, we have done a great deal in trying to understand how the different territories are affected. And we know now that the HFO can be used as epileptogenic markers, because they can be recorded all along the regions that produce and generate the seizures, and can be used to delineate the seizure onset zone. So it is important to try to understand whether all these HFO that can be recording in clinical setting, and that I hope that you appreciate the difference with those microelectrode recording I was showing you before, whether all of them are really HFO, whether all of them are really related with the basic high-frequency oscillatory processes that can be directly linked with specific cellular processes. And here it is important for the clinical setting to understand that we need to establish standards on how to record from these oscillations so that they are meaningful. So we can, clinicians typically work using either the bipolar or the referential montage, but basically they like more the bipolar montage because you average out the noise, which is an important factor in clinical setting. So now bipolar record the potential differences between two active electrodes. And referential montage will record the potential difference between an active and an inactive electrode. And this is the way we typically work in basic settings, so that we don't mix signals. And this is clearly important, because if you have an area producing activity in the brain and you are recording with a bipolar montage, you can be losing activity between two leads, just because you are differentiating between them two. And this will affect the way you are recording and localizing the sources. If you are recording in a referential montage, you will also be able to guess whether you are close to the focus, the region that generates the oscillation, or far away. And this is important because here is an example, a simple exercise, trying to understand the effect of the bipolar and the referential montage using this microelectrode recording from different regions that were separated about 1 millimeter or so. And the waveform of the high frequency oscillation, in this case a physiological ripples in them, is clearly different, although you can clearly identify the positive and negative spiky wave that inform you whether you have a little or more contribution from spikes in these two electrodes. Now, if we take a differential, a bipolar montage between electrode 1 and electrode 2, this is what we get. So we destroy oscillations that were present in the recordings, and we create oscillations that were not present. And to try to identify whether this is related with a particular cellular process, it will be misleading because it will just cannot be identified properly by an expert eye and associated to a particular cellular process. And also the spectrum will be artificially producing or decreasing the main contribution on the frequency band and altering the contribution, the relative contribution between frequency bands. Using filters, also, the most critical example is whether using the standard filters in clinical settings will distort the activity, but also we can use different set of filters to try to isolate the high frequency components from a local field potential recording. But in doing so, we will change the shape of the events. And again, the shape is informative of what were the underlying processes. So here, an expert eye can clearly identify a ripple cycle potentially associated with an IPSP, an inhibitory potential, and here, the spiky component associated with firing. All these will be distorted if we go to filters that evidently will localize the components, the high frequency component, but will lose the ability for us to identify the contributors. And obviously, using these clinical setting filters will prevent us to recognize the complexity of the phenomenon. So now, let's go to the point of what is normal and what is not. I have been trying to tell you whether all these tiny waveforms detail are so important for a basic neurophysiologist to try to understand what underlying processes are related. It's as much important as it is for a clinical neurophysiologist to interpret the EEG and to base a diagnosis based on whether they are recording in the EEG a particular waveform phenomenon. So what is normal and what is not? How can we say what is a normal type of activity or not? These are recordings from epileptic rats. All of these recordings were obtained from the same epileptic rat. So a convulsive seizure, it will be absolutely clear. The animal actually is having this motor activity behavior related with the seizure. So we, for sure, will not have any doubt that this is a pathological activity. We will not get that long in discussing whether interictal spikes are or not pathological, because we know that you never, absolutely never record this very big sharp wave that actually saturate the amplifier events in normal rats. So this is a clear pathological activity. And actually this is what clinical neurophysiology base their diagnostic to determine whether an individual is having or not interictal epileptiform events. But what about these events? What about when we record theta activity in a walking epileptic rat, and you record that from epileptic rats, as you can record normal activity from epileptic patients? And what about when the animal is immobile, that you record the ripple sharp wave complex in these animals? So what is normal and what is not? Is that a normal ripple? Or is that informing that the area is generating ripples, but that the ripples are not normal? So when you record from normal rats with multi-site silicon probes that allow you to record from different layers simultaneously, you know that the high-frequency component, the ripples, are associated, are recorded around the CA1 cell layer, and that they come in associated with a sharp wave that is recorded here around the stratus pyramidal. So this is the sharp wave ripple complex that has a clear, meaningful interpretation of the dendritic activation of pyramidal cells by CA3 and the short-lived oscillations recording in the layer by the interplay between firing from pyramidal cells and inhibitory activity from interneurons. When you record from epilepsy rats, under the same circumstances, you also can identify the sharp wave ripple complexes, but the high-frequency components, actually, in epilepsy rats, are more complex in spectral terms. If you have a look to the time-frequency spectrum of the normal rat, you see that they rarely, this is one event and here is another, with a clear contribution between 100 and 200 Hz. But for epileptic animals, the spectrum is very variable. And one event after the other, the spectral components are different. And this is a major characteristic of this animal. Actually, if you take whatever measure you like from the spectrum, like the entropy, which means how organized or disorganized the spectrum is, and a fast ripple index, which informs you how much of the contribution can be collected at a particular limit, like 250 Hz. So here is a plot of each individual sharp wave ripple complex in a normal rat, black dots, and an epileptic rat. And it's clear that if you take one event, you will not be able to separate that event from an epileptic or a normal rat. But if you look at the distribution of many of them, this will inform you that epileptic animals have a trend to have more disorganized spectrum with larger frequency components. So therefore, what is normal and what is not, pathological ripples have high frequency components that dominate the spectrum, and there is a larger spectral disorganization event after event. This is also the case if you prepare slices from the human hippocampus and you keep them in vitro, and then you use solutions that eventually allow you to induce either in interictal state, where you have sharp waves that recur, or evolve into a seizure-like behavior that you can define a preictal and an ictal state. And in collaboration with Catalina Alvarado-Rojas, who is here, Michel Levan-Cuyen, and Richard Miles and Gilles Uberfeld in Paris, we were examining the high-frequency components of these events. And when you analyze the activity from interictal, you can, from preictal, you can recognize that there are frequency components here that lies within the ripple band, but evidently they should be reflecting something pathological because they are recorded from the pathological, the epileptogenic areas in the human, and also because they are associated with the pathological epileptiform events. Now, if you take all the interictal discharges and the preictal discharges, and you order the spectrum from the more organized to the more disorganized, you see that all of them will exhibit a large variability, and that even you can recognize some ripple-like, very organized spectrum in the interictal, but also disorganized events, showing that this variability is reflecting what is normal and what is not. So what does this spectral variability, why is informative that is a pathological form of activity? Our work in vitro has provided us some cues of how to interpret this. Now, if you take slices from normal rats and you put them in a dish, and you bath the slices with solutions, standard solutions at 32, 34 degrees, and you use between 2.5 and 3 millimolar calcium, you will get this type of activity, which are associated with firing, increase of firing. Many pyramidal cells get inhibited, and some other get excited during these events, and interneurons are firing. Now, this type of activity is quite similar to the one that you can record in a normal rat in vivo. But if you, instead of using 2.5 to 3 millimolar calcium, you use 1 to 1.5 millimolar calcium, which is still in the physiological concentration, the circuits reorganize and the activity that produced now saturate the amplifier. If you are not using a good gain, you can recognize a lot of firing, and actually you cannot sort activity anymore because it is impossible to recognize how they were firing individually here because all cells were firing together. Now, this is a pathological like ripple or oscillation, a pathological like activity, and this is a physiological like activity. And we know that because if you take and start from 3 and go to 1 millimolar calcium, and you record simultaneously one cell and the field, you recognize the high frequency component here, 200 Hz, in this event associated with hyperpolarization of the cell and with a limited neuronal firing. But as long as you change the calcium, this activity will become more and more complex, and the same cells that were hyperpolarizing start to become depolarizing, and activity accelerates. And we show that this is related with a failure of inhibition and a dominant depolarization that underlie the emergence of this pathological type of activity, which are faster. Now, if you take slices from normal rats and you put them in 3 and 1 millimolar, this is what you get. An acceleration of the rhythm, an increase in the amplitude, a lot of cell fire here, which is pathological. So the activity cannot be identified as a physiological ripple because it's associated with an abnormal firing of activity of the cell. So you take slices from epileptic rats and you put it in the dish and you use 3 millimolar calcium, which was the same that using before was associated with inhibition. You see that the activity is distorted, fully distorted. So using 1 millimolar will not help you to differentiate beyond the fact that you have a lot of emergent components in the spectrum and disorganization. But using 3 millimolar will help you to identify clear differences that suggest that there is condition of disinhibition in the slices of epileptic rats. So to make the point simpler, if you are losing inhibition, if you are losing the ability of coordinate the activity of firing cells at the rate that preserve individuality so that not more than 20% of cells fire together during these ripples, physiological ripples, you will get a kind of a type of event which is the ripple, which has a clear spectrum, simple spectrum with single peaks between 100 Hz and 200 and will never invade the high frequency band. But if you go on the conditions of failure of inhibition will prevent the activity to be localized, and therefore they will spread. And because it's spreading in the network, many cells will be firing in phase and out of phase and will be generating high frequency components and larger contribution in the local field potentials. If you take the firing of single cells in vitro and in vivo, and you compare how single cells are firing together with the field, and you plot the relationship between the firing of the cell, the unit frequency of the cell, against the local field potential spectral mode, which is the dominant frequency in the high frequency component, you will see that there is this relationship telling you that when you are recording high frequency components in the local field potential, the single cell will not be firing at that frequency. Using in silico simulations, we previously showed how this emergent component can be related with the increase of synaptic strength and the fact that you are losing inhibition and therefore cells can fire together in phase and out of phase. So pathological ripples emerge from excessive firing, and in phase and out of phase firing from cluster of pyramidal cells contribute to frequency band. So to finish, and I hope that the data can help you to understand what is normal and what is not. Normal is a high frequency oscillation that is associated with an appropriate coordination between excitation and inhibition that keep the level of firing relatively low, although there is an increase, and that is reflected at the level of the local field potential in high frequency component ripples, which never invade beyond 200 Hz band. Pathological high frequency oscillation will be associated with out-of-phase firing. If you take one event might be not different from a ripple event in spectral terms, but you need to take many of them to see how, from one to another, they invade the high frequency band. And this will be informative that the event is pathological. So can we transform physiological ripples into pathological one by altering, for instance, the excitatory to inhibitory balance in vitro? So we took advantage of an integrated fluidic probe that we developed. And this probe, we have tetrodes that allow us to record simultaneously from 4 channels close together, so that can be used to identify individual neurons. And the probe comes with a fluidic channel integrated that allow us to deliver whatever substance very close to the recording site. So using these approaches, we try to test whether we can distort the form of these high-frequency oscillations. I will show you data regarding whether manipulation, the extracellular calcium in vivo, the same we show in vitro, will transform physiological ripples into pathological. So we were recording from the CA1 area of pyramidal cell layer in normal rats, and when we eject EGTA, which is a chelator of calcium, close to the recording site and constrain it to the CA1 cell region, what we observed was that the local field potential waveforms were distorted, and they were transformed in a pathological type of activity that can be recognized because of the spike components and because they invade the high frequency band. And the spectrum disorganized, and what is more importantly, we tested whether inhibition is impaired under this condition by using a protocol which is called paired pulse inhibition, which is the stimulation at an interval of 25 milliseconds that get a full recruitment of the whole population, but when you apply a second pulse, the same intensity, 25 milliseconds after, because it is within the inhibitory component, will give you a lower spike, population spike. And the change in the decrease here informs us that inhibition was affected. So to finish, the pathological high-frequency oscillations represent a very wide band from 80 to 100 Hz, and many different processes that occur at the intracellular temporal scale, from inhibitory postsynaptic potential to excitatory postsynaptic potentials, to high-frequency synaptic activity, to action potential bursting, and many processes can affect the dynamics of the intracellular and the extracellular scale. And there is some linearity in a particular frequency band that allow us to relate a particular phenomena in the local field potential with a particular intracellular process. But beyond a particular frequency, which typically is 200 Hz, there is a breakdown of the relationship because intracellular temporal scale can never go so fast. And when you get activity into this high frequency band, this is not reflecting a single cellular process, but a myriad of events, and eventually it's coming to a pathological conditions. So to finish, and after this discussion, what we can propose actually in this review was a set of recommendations to try to make clinical and basic research studies comparable regarding high-frequency oscillation and their impact in epilepsy. So for HFO, they may reflect oscillatory events and broadband spectral leakage. So we need to check carefully what we are talking about, because not all HFO are the same cellular process. They can be detected with micro and macroelectrodes, but we should be aware that we need to use minimally injuring high-density microelectrode arrays in order to have meaningful information from the human recordings that could be related to what we know from rodent physiology, so that we can identify similar cellular processes in one and another. Using referential montage and wideband filtering is recommended. Later, you can use offline filters to try to segmentate activity, but basically, it's better to record wideband. The event-to-event variability is informative about this, and statistical analysis will be helpful in trying to identify what is normal and what is not in these oscillations. So thank you very much. I'm happy to take questions.
We've probably got time for one question while Catalina comes and sets up. Any questions? Yep, Greg?
Is that, one of the problems always seems that, of course, it's the same networks that are generating seizures that are laying down episodic memories. It's the same structures, same network, same cells. And so would you expect that a region of the hippocampus doing a normal physiologic task, say, consolidation of memory during slow wave sleep or something like that, are those events going to look pathological by the measures that you outlined here, even though it's underlying a physiologic phenomena in somewhat pathological tissue? I hope that doesn't sound too philosophical.
Yes, absolutely. And that is why there is a comorbidity, a cognitive comorbidity in addition. And maybe other diseases that we have not been related to the fact that it is the same circuits, the one that produces the seizure, the one that is operating during a consolidation. For instance, with the ripples, I have shown you ripples recording epileptic rats. And they look slightly different to those recorded in normal rats because of the spectral dissonance, as I tried to show. But the animal is doing exactly the same. The animal is running a task, a memory task. They have to recognize objects, they get to sleep, and do that. So, whether the memory traces were correctly stored or not, because of those processes are altered, for me, the answer is absolutely yes. So, we need to prove it causally, but correlation for sure, we have it, you have it, we know that there is correlation. Because it is the same sequence. So actually, this is my intention is that the cognitive comorbidities, the basis of cognitive comorbidities in epilepsy, are related with the fact that the same circuits are responsible for the two seizure, which is the tip of the iceberg, but also all the cognitive processes. So there is no magic here. I mean, the cell is there. The cell is there participating in a charcoal ripple, in a consolidation process. The cell is there participating in a seizure. In the same cell.
I think we'll have to skip Steve, so you can ask Liset